I'm Amulya and I'm a first year master's student from the Department of Electrical and Systems Engineering. I'll be talking about electron beam microscopy in this presentation. Without microscopy, there's no modern science. Indeed, scientists can't study what they can't measure. They wanted to see more than what the naked eye would allow. Microscopes have come a long way since their first version of a simple magnifying glass that was introduced centuries ago to what we now have, such as SEM, TEM, and other advanced microscopes to view structures of viruses and proteins in 3D. Isn't it a tremendous progress? This progress we are referring to certainly revolved around one parameter, and this person, let's call him Adam, says it's resolution. And it's true. Resolution is the ability to distinguish between 2 points. If we cannot resolve them, they'll be observed as a single point. Resolution of a light microscope is limited by the wavelength of light. This concept of resolution is why we cannot observe the phospholipid bilayer structure of a cell membrane using light microscope, but interestingly we can do so using an electron microscope. Resolving power of electron microscope is much greater than that of the light microscope. The drawbacks of optical microscopy, such as poor resolution, poor surface view, and low magnification, limited their use for various applications. For the rest of the presentation, I am going to be talking about electron beam microscopes, their types, and some interesting applications. Electron microscope was first introduced by two smart folks, Max Knoll and Ernst Ruska, in the year 1931 to overcome the barrier to higher resolution. This relies primarily on transmitting electrons through a specimen instead of light to form an image. These electron microscopes come in a variety of flavors, but the first one to emerge was transmission electron microscope. Quantum mechanics dictates that all particles have a de Broglie wavelength lambda is equals to h by p, where h is Planck's constant and p is the momentum of the particle. For example, an electron that has been accelerated to 0.78 times the speed of light has a de Broglie wavelength of 2 picometers, which is about 100 times smaller than the typical interatomic distance in a solid. Moreover, electrons are charges that can be deflected by electric and magnetic fields. Combining such electromagnetic lenses with an electron gun with some sort of a detection system, we have an electron microscope. Let's talk about what happens when electrons interact with matter. Two types of interactions occur, namely elastic and inelastic. In elastic interactions, the electrons just pass through the sample without any interaction. Therefore, no energy transfer occurs between the sample and the electrons. And the signals originated in this will be utilized in TEM. On the contrary, we have inelastic interaction where energy transfer occurs, as a result of which the energy of the electron gets reduced post-interaction. The different signals obtained in this are predominantly utilized in analytical methods. Transmission electron microscope can be compared to a slide projector, but instead of a light source, we have an electron source, such as tungsten or lanthanum hexaboride or even field emission gun. The glass lenses are replaced by magnetic lenses and the projector screen is replaced by a fluorescent screen. The whole trajectory from source to screen is under vacuum. When electrons impinge on the sample, some of them are absorbed as a function of thickness and composition of the specimen, while some are scattered. The transmitted electrons are counted and sorted to obtain information about the specimen. Resolution of transmission electron microscope can be described in terms of classic Rayleigh criterion for visible light microscope. Momentum is given to the electron by accelerating it through the potential drop V and giving it a kinetic energy EV. By substituting these in the lambda expression, we get a relationship between wavelength and accelerating voltage V of the microscope. We can arrive at a conclusion that if we desire high resolution, a decrease in the wavelength needs to be accomplished by increasing the accelerating voltage. TEM yields information about size, shape, and arrangement of the particles which make up the specimen, along with the atomic scale defects and the elemental composition. There are two types of imaging modes in TEM. In bright field mode, an aperture is placed on the back focal plane of the objective lens such that only direct beam passes. So in this, image results from a weakening of the direct beam due to the interaction with the sample. Therefore, mass, thickness and diffraction contrast contribute to image formation. On the other hand, in the dark field mode, aperture is used to block the direct beam and one or more diffracted beams are allowed to pass the objective aperture. These diffracted beams interact strongly with the specimen, so we can obtain information about planar defects or particle size. Here's an example. The same area of microcrystalline zirconium dioxide is imaged using bright field and dark field mode of TEM. Some crystals appear with dark contrast because they're oriented parallel to the zone axis. The one on the left represents a bright field image and on the right it represents a dark field image. Areas that are close to the edge are thinner and hence they appear brighter than those far at the edge. In dark field image, some of the microcrystals appear with bright contrast, namely whose diffracted beams partly pass the objective aperture. The limitations of TEM are as follows. Aberration limits the image resolution. In spherical aberration, the power in the center of the lens differs from that at the edges. This is largely determined by the lens design and manufacture. In the case of chromatic aberration, the power of the electromagnetic lens varies with the energy of the electrons in the beam. Chromatic aberration can be reduced by keeping the accelerating voltage as stable as possible, which in turn requires sophisticated electronic circuitry. And we also need thin specimens which are less than 20 nanometers or so for imaging. Moving on to scanning electron microscope. Like TEM, SEM consists of an electron optical column, vacuum system, electronics and software. The secondary electron signal is the most frequently used signal. It varies with the topography of the sample surface. It is to be noted that the accelerating voltages in SEM are of the range from 50 to 30,000 volts, which is very low when compared to what we need for imaging using TEM. Different signals originate from different volumes of interaction. Backscattered electrons come from deeper regions of the sample. They are reflected back after elastic interactions between the beam and sample. Secondary electrons, however, originate from the atoms of the sample, particularly from surface regions. SE imaging can provide more detailed surface information. Backscattered electrons show high sensitivity to differences in atomic number. The higher the atomic number, the brighter the material appears in the image. We also have characteristic X-rays. When an electron from a higher energy level drops to fill the electron hole in a lower level, the difference energy might be emitted as high energetic electromagnetic radiation in the form of an X-ray quantum with a characteristic energy. X-ray microanalysis uses an energy dispersive X-ray spectrometer to count and sort characteristic X-rays according to their energy. The resulting energy spectrum exhibits distinctive peaks for the elements present, with the peak heights indicating the element's concentration. Let's talk about some of the limitations of SEM. Many specimens can be brought into the chamber without preparation of any kind. If the specimen contains any volatile components such as water, they must be removed by a drying process before they can be used in a high vacuum system. Non-conducting specimens will accumulate charge under electron bombardment and may need to be coated with a conducting layer. It can be examined without a conductive coating only if the instrument is capable of operating in low vacuum. Most SEMs use a solid-state X-ray detector. And while these detectors are very fast and easy to utilize, they have relatively poor energy resolution and sensitivity to elements present in low abundances when compared to wavelength dispersive X-ray detectors. Scanning transmission electron microscopy is another type that combines the principles of TEM and SEM. Its primary advantage over conventional SEM imaging is the improvement in spatial resolution, which results from eliminating the electron scattering that occurs in bulk specimens as the beam electrons penetrate into the sample. Like TEM, it requires thin samples and it also uses a finely focused beam of electrons across the sample in raster pattern. An advantage over TEM includes the use of other signals such as secondary electrons and characteristic X-rays to obtain information about the specimen. Okay, so we talked about different microscopy techniques. Today, electron beam microscopy is used to study ultra-fast processes, magnify specimens to atomic scale resolution, and measure nanotechnology functionality. It is used at the cutting edge of quantum physics research today. I would like to mention some of the very interesting developments in the electron microscopy in the recent times that have opened the borders for study of structural dynamics at nanoscale. The major challenge with microscopy, even up to recent years, was recreating the fuzzy 2D images as sharp 3D structures. A technique for generating a 3D structure of the protein at an atomic level using an electron microscope involves vitrification to cool the sample to cryogenic temperatures, thereby allowing the biomolecules to retain their shape in vacuum. This approach is called cryo-electron microscopy. This is the image of the birth of a carbon nanotube from a cobalt ferrite nanoparticle obtained using a thermoscientific cryo-TEM. Liquid electron microscopy is an approach to image and examine liquid or liquid-containing samples. It has occupied an important place in molecular research. Various cells and cellular structures can now be examined through thick liquid arrangement. Connectomics is a field that aims at fully deciphering the circuitry of the nervous system on a large scale by mapping every single component and tracing every connection of a certain volume of brain tissue. This work demonstrates the idea of parallelizing image process. This picture represents a coronal mouse brain section from one hemisphere where, with a 61 beam SEM at 4 nanometer pixel size, they were able to acquire 290 GB of image data in 20 minutes. The left image shows the signal electrons in the form of 61 secondary electron spots at the detector. This field of electron microscopy is quite healthy, but focused effort could get it in better shape. The blurriness is from aberrations caused by imperfections of electron lenses. The deadliness is related to the damage that electrons can impart in biological samples. The slowness concerns the availability of electrons to capture atomic behavior that happens on attosecond timescale. It should be noted that femtosecond electron microscopy has been around, but atomic scale spatial and time resolution is still out of reach. To tackle the challenges mentioned, we'll need innovative ideas. So most microscopy applications have mostly relied on classical treatment of the electron-matter interaction. Electron microscopes can show remarkable detail. However, exploiting a particle's quantum nature can reveal novel information not accessible with conventional schemes. An Australian research team has worked on a quantum microscope prototype based on spin sensors. When a laser is shined at the impurities, they emit light that is directly related to interesting physical properties like magnetic field, electric field, and chemistry near the defect. The authors have reported the resolution of their quantum microscope limited by diffraction of light to around 1 micrometer in their results. But they add that this could be sharpened to around 10 nanometers in principle. At this point, it's no longer microscopy but nanoscopy. So there's a huge potential for this new generation of quantum microscopy. These are some of the resources that I used to make this presentation. I hope this was informative. Thank you.